So now I'd like to introduce to you Lori Grace, who will introduce to you Dr. Don Hubbert.
I am a resident of Maui some of the time, and also in California. I work to promote the labeling of GMOs. In the process, I met Don Hubbert and Bob Streit, and I began to learn about the amazing amount of suppression of scientific research and information occurring in the United States. And I began to see that when reporters are writing in good faith in their newspapers, that they are operating like horses with blinders on that cannot see everything. And when I began to discover that the majority of experiments on humans to evaluate the conditions that GMOs create in humans were done outside this country, I could only find one in this country, I began to feel really concerned. Additionally, when I heard about the stoning of scientists Argentinian scientists going to a conference with information about GMOs to report in a conference while they were in their car at a stop sign, or an armed man coming into an Argentinian woman who had lost her daughter to kidney disease and saying, "You lay off these soybeans if you want your 2 remaining children the following morning." When I began to understand these things, I began to feel horrified. And when I gave a grant to support the research on this organism that can't be killed by ordinary means, this organism that can be involved with creating infertility, miscarriage, autism, chronic bowel inflammation, and all of this, when I began to learn about this, and when I created a grant, and the scientists here in the United States were too frightened to let me even know the name of the scientist, I said, oh my God, what a country we're in. What a country we're in. And so, in the spirit of the country that I used to believe in the spirit of freedom of information that I actually believed in when I was a little girl, so proud to be an American, I am bringing over Don Hubbert and Bob Streit to begin to tell you the stories that have not been told, the stories that have been sequestered to Europe, where they do know. And lastly, also, when I read that 100% of Germans who do not eat GMO foods tested positive for Roundup in their urine at 5 to 20 times European levels, and when I began to wonder about Americans and what we, or even the people in Maui, where Roundup is used very heavily, I decided to invest in testing select people, or testing whoever in Maui for Roundup, a limited sample, I have 20, because I really want people to know, and I even want some of our political officials to know internally what might be going on, and perhaps that will motivate and stimulate the courage to take a stand if one knows that you're peeing out herbicide in your urine and your children's. But aside from that, after my passion is over like this, I wanted to now introduce Don Hubbert, who I've known since 2012. He's had 55 years of scientific research and study in the area of plant diseases, and also bioterrorism and how to stop it, and this kind of thing, and has studied all sorts of chemicals. And Bob Streit is with the Iowa Agronomics and works to look at how can we remediate our soils, how can we remediate our bodies also. So both of these scientists are here today, and I want to welcome Don Hubbert to come forward and share some of his most precious information. Thank you.
Lori, I thank you for the opportunity, for the invitation to come, and for the Shaka movement, for the opportunity to participate with you tonight. I'd like to share with you, if we could have the slides. I'd like to share with you just a little bit of the over 1,300 peer-reviewed scientific articles that send up red flags or that absolutely establish that the GMO process and the products that are produced or the chemicals that are used for these chemical-containing crops that are created are not safe. I know of 0 peer-reviewed published studies which establish the chronic safety of any GMO crop or of the glyphosate that over 85% of them are engineered to tolerate. 0 on the safe side. Over 1,300 now that say we need the precautionary principle or they should be banned immediately. This is a mural, Los Angeles, on the side of a cafe that depicts some of the concerns that are expressed in those 1,300 peer-reviewed scientific papers that ask us to be more concerned and to exercise the precautionary principle that each of you are asked to do, not just for you, but also for me, for my 42 grandchildren, my 4 great-grandchildren that I have a concern for, as well as for all the rest that are at stake. We need to remember that in agriculture, we're not dealing with silver bullets and Stinger missiles, as you'll read in the magazines. See the color pictures. Farming is the management of an ecology. It's an ecology that's encompassed by the plant, by the physical environment, and by that very dynamic biological environment that we call the soil. And then you have your pests, your diseases, and those factors that are also involved in production agriculture. But farming is the management of that ecology, managing those interactions so that we favor the plant and we suppress the pathogens and the pests. You can't do one thing in this ecology without changing the interaction of all 4 of those components. They're all based on nutrition. All of the physical stresses can be remediated through nutrition, maybe not completely eliminated, but dramatically remediated. All of the diseases can be reduced in severity. A healthy plant will be resistant to most pests and most diseases, but it's based on the interactions of those 4 components as they interact with nutrition. When we have a compound that is a strong chelator, it changes the relationship of those nutrients in all 4 of those capacities. We have a compound that's unique among all of our herbicides. It was patented in 1964 by Stauffer Chemical Company as a strong mineral chelator. Strong chelator because it immobilizes all of the positively charged ions, your calcium, your magnesium, your iron, your copper, your cobalt, your nickel, your manganese, all of those essential minerals that are critical to the proper functioning of the soil, proper functioning of the plants, of our animals, and of ourselves. In 1974, this very strong chelator was patented by Monsanto as the perfect herbicide. It's unique among all of our herbicides because of the broad spectrum chelating activity that it has. All of our herbicides are chelators. They immobilize a specific nutrient, and that's how they turn off those physiological processes. Glyphosate turns off many. We say primarily active on the EPSPS enzyme. That's in the shikimate pathway. You're told that it's safe for animals and humans because we don't have that pathway. The problem is that all of your gut bacteria that you rely on to live with do have that pathway. And so in 2000, Monsanto patented this herbicide as a very powerful antibiotic. Antibiotic that in their patent at 0.075 parts per million kills your lactobacillus, kills your bifidobacteria, kills your enterococcus, and a number of other bacterial species. In fact, if you go through the patent, I don't think they miss a single taxonomic criteria. The problem is when they take out all the good guys that are responsible for our immunity, for vitamin production, for nutrient absorption, they stimulate all of the pathogens, the Clostridia, botulinum, perfringens, difficile, the E. coli, the salmonella, the listeria, many of your viruses, because there aren't any voids in nature. We have a shift in balance, and when you do one thing, you interrupt the interaction of all four of those components in this ecology that we manage when we're farming. It's really a growth regulator. You can't kill a plant with glyphosate in sterile soil because it has to move down into the root system where it compromises the plant's resistance. And then it's the soil-borne fungi that kill the plant as an herbicidal mode of action. It's an extremely persistent product. It's in the organic phosphate group. It's an organic phosphite, a PO3. Makes it very difficult to degrade. Monsanto has lost 2 lawsuits where they've been accused of fraudulent advertising because they claimed biodegradability, they claimed safety, or they claimed environmental benefit. It's a very unique compound, but 85% of all of our genetically engineered plants are engineered only for the purpose of being able to apply this powerful antibiotic and mineral chelator to the food that you're going to eat or the feed that you're going to provide for your animals. We're concerned about the 29 million pounds of antibiotic, the penicillin, the streptomycin, the tetracycline that we use against the pathogens in agriculture, but nobody seems to be concerned at all about the 380 million pounds of glyphosate that's indiscriminately used throughout our environment, that's changing that ecology, all for the worst. It's been a powerful herbicide for a few years until we had resistant weeds. Lost that power not because the weeds became resistant to the glyphosate. They became resistant to the fungi that compromised plant used to be susceptible to. So we have super weeds that are harder to control. We have weeds that are pathogens with increased virulence as the rate of glyphosate was increased to change that soil biology by killing off the natural biological controls that would control those soil-borne pathogens that were doing the herbicidal activity. And so we see that total change in activity of those organisms. We see the compromising of the plant's resistance by shutting down the shikimate pathway. We see the increased virulence through the root exudates that move out into the soil that affect those soil-borne pathogens then that we live with on all the other crops in our rotation in addition to the genetically engineered ones. And we're left with a compromised soil because we no longer have that natural biological control with the pseudomonads and trichoderma and bacillus and those other organisms in the soil that would help us in that crop production. We have over 40 diseases. As a member of our Threat Pathogens Committee for over 40 years, we used to consider effectively controlled through our management options that are no longer effectively controlled, and we refer to them as re-emerging diseases. Many of them we don't have effective controls for. Some of these diseases are new. Corn osprey root rot on soybeans. Number 2 disease in Brazil. I've seen 40% yield losses in the Midwest. Yet 30 years ago, this organism was considered only a soil saprophyte. But you put a little glyphosate on the leaves of a Roundup Ready soybean plant, it changes the soil biology, and this normally harmless fungus all of a sudden becomes a virulent pathogen. We have genetically engineered crops, and you say, well, doesn't that take care of these chemicals that we're putting on them? You have to remember that there's nothing in the genetic engineering technique that does anything to the chemicals that we're applying. All it does is make the plant tolerant so that it's not killed by these soil-borne fungi when the chemical is applied. The chemical, glyphosate, as it's applied, is still a strong chelator. It's still compromising that soil ecology that we need for the overall health of our soil, for the aggregation and for the porosity, so that we can receive the water as it's given to us in the rainfall and have it penetrate rather than have it run off as we're seeing on so many of our soils now. The other thing that you have to remember is that 80% of that glyphosate is going to stay in the plant for the life of the plant. As it moves around, it's systemic. It accumulates in the root tips, the shoot tips, the reproductive structures, and your legume nodules. When it's used as a ripening agent, the only place it can accumulate, the only sink that's left, is right in the seed, in the reproductive structure. The EPA has had to repeatedly increase the tolerance levels that we can have, and they said, so we have safe food. Not based on any scientific studies, because the recent levels that they raised are 4,000 times the level that has been documented to inhibit the endocrine hormone system. Over 4,000 times the level that's required to change the estrogenic and oestrogenic hormones. You see that entire system changed. And so in the first 3 months of gestation, which is so critical in the development of a child, you see cleft palates resulting. You see purpura. You see the anencephaly. You see the many other conditions that are occurring in our children. and children worldwide where this chemical is so indiscriminately used. Causes a yield drag. We're told that we can't feed another 2 billion people unless we have genetic engineering. We don't have a single plant that has increased, a genetically engineered plant, that has increased the intrinsic yield of any crop. Yield and quality are multifactorial conditions that we don't have the knowledge with our primitive understanding of this genetic engineering process to accomplish. Our genetic engineering, as it's practiced now, is more like a virus infection than it is a normal breeding program. You even have, there's nothing equivalent with hybridization or normal breeding programs at all. You're throwing in genetic material. It even requires a cauliflower mosaic virus in order to activate it, to promote it. You have all your antibiotic genes, resistant genes. You've changed the entire system. Again, it's like a virus infection. It's fossil science. We had to throw it out as our understanding of genetics when the human genome was sequenced. The Scientific American article said, where are all the genes? Our concept with genetic engineering is that we have a gene for each function. That's 30-year-old science. Our understanding of genetics now is that it's a spatial relationship influenced by the environment. When you throw in genetic material, even as they said when they genetically engineered soybeans to have resistance to 3 different herbicides, they said we only had to add 4 base pairs. Takes about 50 base pairs for a gene. Didn't have to do very much. Changes the spatial relationship. Then they said about 15 pages back, oh, but we had to delete 4,383 base pairs. But they said we think these are fossil genes because it didn't seem to kill the plant. Now, they may have killed 2 million of those cells and only had a few cells that were harvested. It's fossil science because we know that it's a spatial relationship. There are thousands of epigenetic factors and mutations, teratogenetic effects that have never been analyzed for toxicity. Even the genetically engineered plants are compromised. As you can see here, less of the structural materials in the plant, the lignin. Look at what happens to photosynthesis up on your far right up there. That's why we're growing the plants, so that we can capture the sun's energy and use that energy then to fix the carbon dioxide with the hydrogen out of the water that's split with manganese and form sugar that we can store and use. You see also the drop in amino acids. That's what we're feeding. That's where we get some of our essential amino acids over here. Compromised nutrition. And then we talk about climate change and its impact on agriculture and us. It takes twice the water in a genetically engineered soybean plant to produce a pound of dry matter when it's treated with glyphosate than its parent without the glyphosate. Greatly compromises our sustainability, our resistance to many of these abiotic challenges that we have. And what happens long term as we see the accumulation of some of these persistent materials such as glyphosate. Our growers will say, my crop doesn't look as vigorous as it used to. They'll blame it on the seed. Part of it is from the seed because they're of lower micronutrient content that would help buffer against that. But it's also that change in the soil biology and the change in the ability of the plant to achieve and to obtain those essential nutrients. We see that nutrient deficiency is greatly increased in all of these plants. We see all of the other feed and food safety issues now that are being finally published. Difficult to publish if you're talking about genetic engineering or even the glyphosate or glyphosate types of herbicides. We're finally getting some of those published. There's been tremendous scientific censorship. We saw it with the Cirolini paper just recently, 16 months after it was published. One of the most extensively peer-reviewed papers that we've had, the only long-term study on the genetically engineered proteins for Roundup Ready and the Roundup herbicide that's used. I'll show you a little bit of that response. You see all of these factors that are now involved. We talk about an obesity problem. We talk about mineral deficiencies and increased nutrient susceptibility for our animals and our children and ourselves. This is Roundup Ready alfalfa. Look at what's compromised in that nutrient density. Sulfur, that's your methionine, your cysteine, your resistance mechanism, your immune system. All of your proteins start with methionine. Look at what the compromising is. Look at our manganese, a third less. Every animal that has been necropsied at Iowa State's large animal laboratory the last 2 years has shown a critical deficiency of manganese and vitamin B12, which is cobalt. Both of them because they're chelated, immobilized by this powerful chelator and antibiotic that is so insidious in our crops and our feed materials. You see in Roundup Ready soybeans, we've compromised the ability of that plant to capture the sun's energy and store it as sugar. But then look at what happens in that material that we feed our animals or that we rely on also for our minerals. And you see that compromising taking place. You say, well, does it have an effect? It's a study of Dr. Jeffrey Shepard's University of Minnesota. Had a 5-year study trying to figure out why there were so many stillbirths, why so many calves were deformed. 100% of them deficient in manganese. 15 years before, we thought we had too much manganese, recommended that they cut it back in the ration. At the same time, we introduced Roundup Ready crops, corn and soybeans. Now we have Roundup Ready alfalfa also. You see the consequences, the same consequences in our children when they don't receive that proper nutrition. Animals can tell the difference. Those 2 ears at the top are isogenic lines. The only difference, one is genetically engineered, the other one is the parent that wasn't engineered. Those 2 ears stayed in the tree for 9 months. The squirrels had a preference for which one they ate. You look at those 2 bags again of isogenic corn, triple stack, non-isogenic parent. As Gilbert Hostetler said, maybe the mice know something that I ought to know. Before my presentation in Florida a month ago, I had a farmer come up to me, wanted me to know that he was all in favor of genetic engineering and that he was going to be present and listening to me. His comment was that I grow commercial sweet corn, have for 30 years. Said I went Roundup Ready sweet corn this year, and it's the first time the coons wouldn't eat an ear. So you're going to feed it to your children. What we're finding is that 80% of the genetically engineered corn contains from 10 to 200 parts per million formaldehyde, a known carcinogen, zero tolerance. In 2002, we had the head of the United Stock Growers Association that went to Congress, gave testimony before the Senate Agriculture Committee. So there are two conditions that are threatening the survival of our industry. One of them is premature aging. Take a prime beef to market, grade it down as like it's an old cow being culled out of a dairy. The second one was infertility and reproductive failure. CDC just told us that this generation of children coming up now will have a shorter life expectancy than their parents. It's never happened before in the history of medicine. Where's our technology? It's the same thing that the United Stock Growers had recognized in their cattle. We had a study 4 years ago in Iowa, and you can see the effect of GMO feed versus the non-GMO with that yellow tallow. That's an allergy response. If you look at the stomach and look at the intestines, you'll see the ulceration. You'll see the deterioration of the intestinal lining. You'll see that intense allergy response from these foreign proteins that are now being produced in our food and in our feed products. Paper published in the Hordes Dairyman, November 2011, addressed the second issue. That was reproductive failure. They surveyed dairies from Mexico to Canada, all over North America. This is some of the data that they published in that article. It said, why are so many cows losing pregnancies? We have dairies that have lost up to 70%. The United Stock Growers stated in their testimony to Congress that it wasn't unusual to have 40 to 50% lost pregnancies. And then you read the USA Today, and it said a few months ago, birth rate, fertility levels in the United States dropped another 30% in the last 5 years. That's on top of 25% the previous 5 years. There are some veterinarians that were concerned about this because we had some high-value animals that had lost their offspring, miscarried. They were told by the owners that if you want to keep your job, you find out what the cause is, because they could rule out the botulism toxins, the mycotoxins, all of the other known causes of infertility or of miscarriage, and yet this problem was continuing to accelerate. In 2005, we had one veterinarian that had exhausted all of the other information that he could find and decided to go to an electron microscope to see if there were physiological changes that might be giving him some clue as to what was happening. What he observed, and he first observed it actually in 2002, was a new entity. It was considered in 2002 as a suspect agent because he always found it in amniotic fluid, in placenta, and in aborted fetal tissue. As he started studying and working on this, he was finally able to culture it, to get it to grow in sterile culture in 2005. Initially thought to be a new life form because of its ability to self-replicate. He could grow it before that with a nurse organism like we have to do for viruses or phages or a number of other very fastidious organisms. But he was able to grow up enough that he first isolated it from aborted fetal tissue, grew it up in culture, reintroduced it to the animal, reproduced the symptoms, and re-isolated it to go through that scientific procedure of Koch's postulates to establish that this was the cause of the reproductive failure. Causes infertility in male and female, causes miscarriage in cattle, horses, sheep, pigs, poultry, and people. It was initially referred to as a microfungus. It's a misnomer. It's not a living entity. It doesn't have DNA or RNA, but it self-replicates. There are two other possibilities. If you look on the right, when Dr. Michael Antonio at King's College, the world's authority on prions, looked at this entity and he said, it looks 95% like a prion. A prion doesn't have any DNA or RNA, but it self-replicates. Very important in the brain as long as you have copper as that transition element to hold it. You upset that relationship and you have mad cow disease. You have free prions. It's also known to be very important in Lyme's disease and several other diseases. There's another entity, though, that also self-replicates. Doesn't have DNA or RNA, and it's called a biomatrix. Initially, it was thought to be a new living entity. It was called a nanobacter. First discovered just in 2000. In 2010, they finally realized, after even giving it a taxonomic name, Latin binomial, that it wasn't a living entity, didn't have DNA and RNA, but it's a nano-sized crystal. Then a biological fluid self-replicates, and it grows and grows. But it has a unique characteristic in that it has both an anion and a cation chelating ability, so that it can take the free amino acids in that biological fluid, whether it be in your blood vessels or your arteries, or whether it be in a joint of your body or some other biological place where you have those free amino acids then that are chelated, and it forms a protein coat or protein strands, very much like a virus does its protein coat, like you see with tobacco ring spot virus coat protein in papaya. However, with this biomatrix, it becomes a filtering entity, and it will continue to grow as long as there is substrate for that nano-sized crystal to develop. When that stops, then it implodes with all the protein, with all the other material that's been entrapped, and if it's involved in atherosclerosis, then's where you have a massive heart attack that the patient rarely survives. 3 months after my letter to Secretary Vilsack asking him for help and to release the muzzling of the USDA scientists that had indicated a desire to work with us on this entity and with the veterinarians, the Chinese sent me an email, had 4 electron micrograph slides, and said, "Is this your new organism?" It only took 3 months. Sent it to the veterinarians, and they said, "It appears to be." But they said, how in the world did they ever get such a clean culture and such heavy growth on it? I emailed back to the Inspections and Quarantine Division in the China system. I said, what media are you growing it on? They emailed me back and they said, we haven't been able to grow it like you have. All we did was go into the bin where we'd imported some soybeans from the United States. We planted them in the greenhouse, grew them up to the second trifoliate stage, and merely took some scissors and then put that cut leaf surface on an electron microscope grid, It looked like you'd taken the top off a 5-pound bag of sugar and poured it across the table. Our veterinarians, once they had established that this is the cause of the reproductive failure, said, how are the animals becoming infected, or how are they getting this entity? Soybean meal in the feed was just loaded with it. We find it in corn. We find it in silage. Any place where we have the GMO feed or crops with high glyphosate usage, or where we've had a GMO product and high glyphosate usage, you can find it induced in other plants. We're still in that last debate of, is it a prion or is it a biomatrix? It has characteristics of both. This is a study published in September of last year, the only other long-term study on GMO crops. Study in Iowa with pigs, 5-month study from weaning to market. These are what the stomachs look like. They're very close to what your stomach looks like if you're eating GMO crops or not. Gotten off on a tangent here and need to move. What do we do? What do we do if our children can't tolerate breast milk or cow's milk? We put them on soybeans, 98% Roundup ready. Let me move through some of these others. This is a study from Shaka and the group, poultry microbiota. These are the good guys over here that glyphosate is toxic to. Look at the ones that glyphosate stimulates that we worry about all the time because of the extreme health factors that are involved. Study by Dr. Monica Kruger in Germany. Severe epidemic of chronic botulism. Every single case of sudden infant death syndrome where they've been using the new test strips for the bot neurotoxin, thank you, has shown that it's chronic botulism. What's the symptom of sudden infant death syndrome? Respiratory failure. What's the symptom of the bot neurotoxin? Respiratory failure. Doesn't kill the cow outright. In humans, we call it chronic fatigue syndrome. And you see that relationship when those organisms that would normally control the Clostridium botulinum are taken out by 1/10 of a part per million of glyphosate in the feed. Then you see the botulinum, Clostridium botulinum developing and the bot neurotoxin developing until the cow finally dies. Why do we have 50% of our honeybees in the Western world where we use extensive glyphosate dead from chronic or from colony collapse disorder? It's an extreme endocrine hormone disruptor. But what really kills the bees is it takes the lactobacillus and bifidobacterium out of the honey crop, out of its intestinal system, so it can't digest the honey and the protein that's still in the hive. You look at all of the direct toxicity and the levels that it takes for end-stage kidney failure, for any of the other diseases. And then you see that 40 parts per million glyphosate is considered safe by EPA in refined soybean oil. 400 parts per million in alfalfa. Look at the curve. This is with celiac disease in children that's severe enough to require hospitalization. Follow the curve from the incidence of GMO canola being released, also the time when desiccant use of glyphosate could be used to dry down the grain. That same curve fits for every one of these diseases, all of them approaching epidemic status in the United States, many other parts of the world. You say, what's changed? I served for 8 years for the Surgeon General on the quadripartite Global Epidemiology Committee. If we'd seen that curve on 2 diseases, would have assigned a scientific panel, given them $5 or $10 million to find out what changed. Here we have 32 or 34 that we still say can't figure out what's happening. Samsonov and Seneff at Massachusetts Institute of Technology recently published their second paper connecting all the biochemical dots with glyphosate and the GMO crops with all of these diseases. What's changed? Dr. Nancy Swanson at Seattle plotted the CDC data for those diseases. You see that time when either glyphosate was introduced in '74, and then that tremendous jump for all of those diseases when we went with the GMO crops and greatly increased the amount of exposure to glyphosate because these crops are designed to tolerate it. The Seralini study, the kidney failure, the liver failure, the onset of cancer, all of the other problems because of the extreme toxicity, not just of the glyphosate, but even with the protein, the foreign proteins on their own. National Cancer Institute annual report. All of those red lines up there are target tissues for glyphosate. And then you look at all of the other studies and the anecdotal evidence that we're seeing from around the world. Australia, Argentina, 447% increase in children's cancer. And then you see all of the other problems that have been involved in the safety, quote unquote, studies for the GMO crops as they've been submitted to the regulatory agencies. You see the fraud and the falsification of data that the EPA scientists have pointed out in their own studies. And then you see all of those systems as they fit together and associated with the deterioration of the health of our soils, of our crops, of our animals, and of our people. To summarize, I would just state that future historians may well look back upon our time and write not about how many pounds of pesticides we did or didn't apply, but they'll write about how willing we are to sacrifice our children and jeopardize future generations for this experimental procedure that's based on flawed science and failed promises, just to benefit the bottom line of a commercial enterprise. I wish you success in your endeavors. You have to do it. Thank you for the opportunity to join with us.
All righty.
We were hoping to have questions and answers, but Dr. Huber has to head to the airport directly, so we're going to skip that phase. And
All right. Testing this mic. Thank you. All right. So I want to try a real quick little exercise because we have about 10 more minutes that we'd like to have your attention. Oh, okay. He's going to stay for 10 minutes and he'll do his plane a little bit later. So call the airport and... Just so you know, real quickly, what you just experienced was a summary of his information. And on Friday, he gave a private lecture for some of the organizers working with the Shaka movement and some other scientists, environmentalists. We filmed the entire almost 2.5 hours, and it'll be available at the Shaka movement website if you want all of the data, all of the slides, all of the information. So we need to, is anyone interested in asking a question? Because what we would like you to do is to come down here to the front of the stage while we do that. And we want to inform you that while people are filing down here to do that, on March 15th, Saturday, March 15th, is a huge rally that we're doing. And we'll have other international speakers there. Our 5 citizens will be there. We have musicians, and it's an incredible opportunity to actually have all the Ohanas together. So I'm inviting you to go out from this moment forward, tell as many people as you can about what is happening, and invite them for Saturday, March 15th to the rally. Come to shakamovement.org to find out the specific time and the specific location. So our first question. Questions, please, no statements.
Can you contact Bill Gates and all those rich people that are supporting GMOs and tell them to knock it off because they're killing the world?
Anyone have his number?
I'll need that repeated.
You wanted to know if you could contact Bill Gates and tell him to stop funding GMO.
I wish I could. I should apologize here that I can't stay very long, but and I usually move around. At my age, that's the only way you can tell the difference between rigor mortis and arthritis. But
I'm not sure who this question would be asked to, but does Monsanto have anything in place for future, like down the line, if we do find out that these are causing cancers, will there be people be compensated financially in lawsuits?
when a farmer buys seed, and it's really interesting, when a farmer buys seed, he signs a statement that he will relieve either Monsanto or Syngenta or Dow or the other developer of any current or future liability that may come from that seed. Now, if the product is so good and so safe, used in accordance with the label directions of the manufacturer who declares its safety to EPA and FDA, why do you have to have that statement in there? I'll tell you why. I have a law firm that estimated what the medical expense account, the trust fund, will be for the damage that's being done today by the GMO products, proteins, and the glyphosate that's applied to them, neither one ever having been properly safety evaluated. They estimate in excess of $200 billion. If the waiver holds, that liability comes back to the farmer who planted the seed. If you read Karen McAfee's economic analysis, Karen's a professor at Yale University, an economist. She says that the only people who have benefited from genetically engineered crops are the commercial company, not the consumer, not the farmer. Those two have actually been compromised in the process, both through the reduced nutrient efficiency, nutrient density of the food, and also by the increased disease susceptibility, which reduces production efficiency of those crops. They couldn't get expression of the Roundup Ready gene or the Bt toxin in corn until they compromised its resistance to Goss's wilt, a systemic bacterium. We had excellent resistance. For 30 years, that disease had never moved from a few counties in eastern Nebraska, western Iowa. It's now everywhere in North America and anywhere that the seed that's grown in the Midwest is transferred. They compromised the farmer and the consumer. We'd have a lot of farmers who wouldn't be in business today if it wasn't for federal crop insurance. which subsidizes the damage that we're seeing from the genetically engineered products.
Yeah. Dr. Hubbert, I know that you have one of the most I know that you have one of the most illustrious careers, and I would like you to explain to this group, you have an incredible resume that hasn't really been spoken about. You have a pedigree that cannot be beaten. And I think that's important that people know the quality with which you bring to this company, our company. Us.
Can you translate that? In response to said that you've had a distinguished career, and where can people find out more about your career? And your
List your career.
I've had 2 careers, been really blessed, loved to farm, couldn't afford to farm. Actually, it was squeezing cows that convinced me to go to college. But had the privilege of serving this country, retired as a full colonel in 1995, as associate professor of the Armed Forces Medical Intelligence Center, served on the U.S. Congress Office of Technology Assessment, also chaired several of those study groups for pesticide alternatives, biological control, for vulnerability. Served for 40 years, I indicated, on the Threat Pathogens Committee. I'm kind of the institutional memory on that. Still keep kicking for some reason. Maybe it's I still have something to do, or as my wife said, I need a little more time to repent. So had the privilege at Purdue University, and that's where we really raised our 11 children. And Purdue was very good to me. Gave me flexibility to do some research. My last 20 years were dedicated to how do we get out of the hole that we keep digging? How do we remediate? And there are some things that we could do. I just didn't have time. Get into those. Bob Strike can share a lot of those things with you. He's doing some great research. But again, better run. It's a long swim. But again, many thanks. Appreciate this opportunity.
So as our 5 superheroes leave the stage, let's have one more round of applause for them. Thank you all.